I should have kept you. I should have tried. I should have been a wiser kind of guy. I miss you. Give me wings. Give me space. Give me money for a change of pace. Those noisy rooms and passing rounds. I love you. Where's the morning in my life? Where's the sense in staying right? Who said time is on my side? I've got years and hours and money in my life, but I'll survive your naked eyes. I'll survive. Across the floor, you and me. I know life until I have to leave mine.
Welcome to Metabolic Physiology in Isolation, Day 8, where we're going to hear from Paul Titchnell from UPenn. I'm here with my co-host Sid.
Hi.
And we're here to get you rolling with Day 8 of Metabolic Physiology in Isolation. I've said this every day, but I'm going to say it again. This is a truly new model for inclusive global science. Anybody can listen anywhere at any time without any cost. The White Magara Lab is very happy to take on that cost. All recordings of every talk are going to be listed both on the White McGarrett Facebook page as well as in YouTube. All you have to do is use the hashtag MetPhys2020 to search for the talks. They're listed under the day of presentation with titles, so you'll be able to find all of them. And that usually takes a couple of hours after the end of the talk before it's able to be uploaded onto YouTube, but they are there. Cassidy, it's your time. You are leading the way with the Inception Challenge. You've managed to take a photo in front of the screen since day 1. We're at day 8, and already that photo is getting pretty impressive. Take another one, share on Twitter. We're all looking forward to seeing that. I said yesterday that the teacup challenge was over. I was clearly wrong about that. There were multiple entrants that came in. I'm just showing a couple here. Gustavo, I have no idea what you're doing with those tea leaves, but it looks delicious. And that goblet is impressive. Why you need to stir them, I'm not sure, but hey, I'd love an explanation. If you can get on Twitter and share that with us, I think we're all a little intrigued. Stephanie, your cup is beautiful. I don't think there's a nicer gold handle out there, but I might be surprised. I bet I'm going to see 30 gold handles today. That's just the way this thing works. And Gareth, we've challenged you to do latte art, but for some reason you've decided to make maple syrup. That was not your challenge. Get back on the job. You do not need all that equipment to make lattes. All right, can somebody get Gareth latte machine? That's what I'm saying. Let's group fund this. Let's get him making some latte art. He needs to make this challenge. We want to see it happen. Sid here makes great latte art, don't you, Sid?
Yeah.
That's right. So if Sid can do it, come on, Gareth. You don't need a PhD. All right. Now that I've made fun of Gareth, who's going to be speaking later, let's remember everyone else's faces as well. These are the people who replied almost immediately as I was trying to put together a program for MetPhys 2020. That's right. This program was not organized prior to the shutdown. We just felt that we could come together as a metabolism community and share the great science everyone is doing, kind of give us some hope, encourage you to stay home and flatten that curve. And I think so far, every speaker has done an amazing job. Not only are they sharing their work, they're sharing their unpublished work, and they are sharing a lot of it. So I know, like you, I'm really enjoying these talks. And today we've got another great one with Paul Titchnell. If you want to thank these people, get on Twitter, ask them questions, tell them how much you like their talk. And if you see them in person, let them know that you saw their talk and you enjoyed it, and that's a great encouragement. I'm sure everyone else will be as pleased about that as I am. I have enjoyed the messages I've received, and that makes me feel truly happy. But if you do want to thank me in person, the way you can do that, that would truly honor me, is to reach out to the American Diabetes Association and make a small donation to the Pathway to Stop Diabetes program. That's the program that funded my program, which allowed me to start my own lab. They're truly funding amazing research. If you go on the website that's listed there, you'll see all the fantastic projects that they are really funding, and this is state-of-the-art science. We have another speaker from the program later in the program, Celine Riera. You're going to hear about her great work with olfactory receptors and how they influence metabolism. It's really interesting. But without further ado, I'm going to hand it over to Paul. I just want to remind you that tomorrow is going to be another incredible talk. This one's, I think, going to be our first one from across the pond. We're going over to the Helmholtz, where we're going to hear from Natalie Kramer on proteomic profiling of cellular organization and signaling in metabolic diseases. This work is really exciting, and I know you're all going to enjoy it, so make sure you tune in tomorrow. And if you can't tune in, get onto YouTube, get onto Facebook, and listen to those talks when you can. They are truly going to be great. All right, so I'm going to hand over the host controls to Paul. Sid and I are going to say peace out, stay clean, stay safe, and we hope to see you again tomorrow.
Peace out, man.
Paul, I'm about to give you the host controls, making host. Yes, I want to give that to you, Paul.
Okay, thank you, Phil, for putting on this incredible seminar series, keeping us all sane in isolation. I just got the okay from Phil. It looks like everybody can see the slides. I'm really pleased and very happy to contribute to this. This has been an incredible week or so of science and in particular metabolism research that I'm really glad that we can contribute to. So thanks again to Phil and the White McGarrett Lab for putting this spectacular series on. Today, I'm going to tell you a little bit about what my lab has been interested in recently over the last 3 years or so, trying to really understand the basic mechanisms of insulin action, primarily in skeletal muscle, with the ultimate goal to try to understand this incredible paradox that we've been struggling with for quite a long time of insulin resistance. So today I'm going to show you for the first half a recently published story in Molecular Metabolism at the end of last year, then in keeping aligned with the spirit of this series, the second half of the presentation will be all unpublished work that I would love to get your feedback on via Twitter, and you can see the handle there in the bottom left of the screen. So without further ado, let's get started. And as I mentioned, my lab is really interested in insulin signaling and insulin action. And for those of you, I know we have a sort of broad viewing base with including some students. I thought I'd bring a little bit of historical perspective about why we're coming and trying to address this question of insulin resistance and where this really question is founded upon. And so in the late 1920s, it became quite clear that certain individuals required sort of really large amounts of insulin to control their hyperglycemia. And predominantly, these were individuals that were either sick with some kind of infection or succumbed to some iron overload disorder. And this is a seminal article put together by Howard Root in the New England Journal of Medicine, where he was quoted to say that such great variations in the amount of insulin required to reduce hyperglycemia were at times observed that the use of the term insulin resistance seemed justified. Remember, this is at an era when Banting and Best and McLeod and Collop really were treating individuals with insulin and leading to curative responses in what we now know as sort of type 1 diabetics. However, there was this large, at the time, relatively small, but growing number of individuals that required sort of this excess amounts of insulin, which led to Root sort of postulating this insulin resistant term. About a decade later, in the Lancet, Hemsworth et al. Did a very simple and elegant experiment at the time to really demonstrate the difference between the two types of diabetics at the time, which we now kind of know of as either insulin sensitive or insulin resistant. And he did a very simple experiment, what we now know is sort of like an insulin tolerance test, where he would inject individuals with either glucose and insulin simultaneously. And these are all, sorry, let me step back. And these are all sort of quote unquote diabetic individuals who had hyperglycemia. If you inject a insulin sensitive person with glucose and insulin, you could notice this robust drop in blood sugar. This person, despite being diabetic, is very capable of responding to insulin, and that he turned these people in turn insulin sensitive. That was opposed by individuals where if you inject glucose and insulin, same concentrations, you see this rapid rise in blood sugar levels over time. And at the time, he postulated that these people were insulin insensitive, and as we kind of more collectively know them as insulin resistant. He initially said insulin insensitive because he hypothesized that they were missing a sensitizing factor to insulin that led to this additive effect. So this is one of the earliest indications that there's two groups of people that, despite both being diabetic, have different responses to insulin. And such, the insulin resistance field has been trying to understand the phenomenon, why and how are some individuals resistant to the hormone insulin. For insulin sensitive individuals, it's quite clear. It's not where my lab is focused on, but several other groups around the world are really trying to understand why the beta cells fail, why those individuals fail to produce enough insulin to appropriately control their blood sugar. My lab in particular is focused on the other set of individuals that's more closely aligned with the obesity epidemic, type 2 diabetes, really to try to understand how insulin resistance begins and how that progresses. So in particular, as many of you know, the skeletal muscle makes up most of the mass of a human being, and as such is a pool for a lot of the glucose that's available into our body. And so early work by several groups, here I'm just highlighting one in the JCI from Kolderman et al., suggested that the skeletal muscle of quote unquote diabetics were insulin resistant. So that makes somewhat sense to us in the fact that this contributes to a large amount of the total disposal pool of glucose. And this is what's called a hyperinsulinemic euglycemic clamp study. So under these conditions, you can infuse insulin at various doses, here in a stepwise fashion, and couple it with radio-labeled tracers to measure the degree of glucose disposal at the body. Now this is the whole disposal rate. And as we know, skeletal muscle makes up a majority of this, with fat and other tissues contributing some. And you can see quite clearly in impaired glucose tolerant individuals, or what we now call sort of pre-diabetic, there is a shift of the dose response curve to the right, suggesting that you need roughly twice or maybe 3 times as much insulin to get you the same degree of glucose disposal. By definition, this is insulin resistance. This is even more dramatic in frank diabetes, with type 2 diabetics that are either non-obese or type 2 diabetics that are obese. And you can see a dramatic shift of the dose response curve to the right, and even a failure to completely stimulate glucose disposal, even at really high supraphysiologic insulin concentrations. So it became quite clear that total glucose disposal in insulin-resistant individuals was impaired. And as I mentioned, the skeletal muscle makes up a majority of that pool. And work by DeFranz and others, this is one of the more recent studies, doing similar experiments in humans, measuring skeletal muscle, now specific glucose uptake. You can see in the forearm, you can see both in obese and then in type 2 diabetics, either lean and obese, a dramatic reduction in skeletal muscle specific glucose uptake. So the defect in the skeletal muscle, since that it accounts for a majority of the total disposal of the body, is what some have hypothesized to underlie the hyperglycemic and insulin resistant phenotype. So for years, once this was known, people have tried to understand where in the pathway is the defect. And some of the initial hypotheses was that it's either a defect in the ability of the muscle to take up glucose, or it is an ability to have a defect in metabolism of that glucose. Work by various groups over the years have really tried incredible efforts to understand the signaling pathways, which we're going to talk about in a moment. But one of the major breakthroughs was the identification of the insulin responsive glucose transporter, in particular what we now know as GLUT4. And once that was identified by James and Birnbaum and others and cloned, several groups have went on to show that there was a defect in insulin's ability to stimulate GLUT4 translocation to the plasma membrane. So my hypothesis could be that there's a defect in getting the GLUT4 to the membrane, or that there's an inherent defect in the ability to take up glucose with transporters located already on the membrane. So sort of this translocation hypothesis was given a lot of support in some of these early studies that demonstrated there was a marked decrease in so this is sucrose density fractionation of GLUT4. Here you can see an insulin stimulated during clamp conditions, insulin sensitive or insulin resistant or diabetic individuals. In response to insulin and insulin sensitive individuals, you see this marked increase in GLUT4 that's associated with the 32% sucrose fraction. So this is the fraction that is most commonly associated with the plasma membrane. And you can see that that shift and that translocation is markedly defective in both insulin resistant and type 2 diabetic individuals. That is sort of quantified as a fold change here. So in insulin sensitive individuals during clamp conditions, there's roughly a 3-fold increase in GLUT4 translocation to the plasma membrane, and that is markedly decreased in both insulin resistant and frank diabetic individuals. So that collectively led to the hypothesis that there's this defect in insulin's ability to stimulate GLUT4 translocation to the plasma membrane that led to a defect in glucose uptake in skeletal muscle. And that in turn leads to a dramatic decrease in glucose uptake and thus hyperglycemia, and sort of insinuating the sequelae of insulin resistance. So here, this is sort of the conceptual basis of insulin resistance. Here in a cartoon illustration, insulin stimulates GLUT4 translocation, as I just mentioned to you in the previous slide. And in metabolic labeling studies by Schulman and others have labeled the glucose and has indicated that 3/4 of the glucose that is taken up by muscle is put into the glycogen, while about 1/4 is oxidized through various glycolytic pathways. And in insulin-resistant skeletal muscle, the sort of hypothesis is that there's a defect either proximal or distal to GLUT4 in its ability to get to the plasma membrane, thus leading to reduction in glycogen synthesis and glucose oxidation. So this is not sort of a new hypothesis. This has been around for a really long time, and people have been interested in this question for quite a while. So the big million-dollar question in the field at the time was, and still remains, is where is the biochemical block in GLUT4 translocation, glucose uptake? As I mentioned, you can hypothesize it's either proximal to telling GLUT4 you should get to the membrane, or you could tell GLUT4 you should get to the membrane, but there's somehow an inherent block in its ability to do so. So investigators all around the world for several years, decades, have been trying to elucidate the signaling mechanisms that are responsible for skeletal muscle glucose transport. And in particular, focus has been on the signaling mechanisms, the proximal mechanisms, which I'm really going to highlight here, what the rest of the talk is primarily about. And as many of you know, insulin signals through the insulin receptor, which is a receptor tyrosine kinase that leads to the activation and recruitment of insulin receptor substrate proteins. This, in turn, leads to the activation of PI3 kinase and subsequent activation of the serine threonine kinase, AKT, or protein kinase B. And supportive of that hypothesis, many have wondered, is this pathway involved in telling the skeletal muscle to stimulate glucose transport in response to insulin? So several studies here, I'm going to highlight a couple. One is by Laurie Goodyear that has indicated that in obese, insulin-resistant human muscle biopsies, if you look at IRS activity, you can measure here in basal or with multiple minutes of insulin stimulation, you could see a marked decrease in IRS activity. And if you do a PI3 phosphoinositide blot looking at radio-labeled PIP3, you can see that in addition to IRS defect, there's also a defect in PI3 kinase activity. Okay, come on in. Sorry, we have a guest. Sorry to interrupt. Come on, my daughter, Tori. She was interested in insulin signaling, so she figured that she'd come and listen. Come on, sit over here. In addition to PI3 kinase, downstream, most distal to that is AKT. And you can see here's just a kinase assay looking at AKT activity using GSK3 peptide. And you can see in insulin-resistant, non-insulin-dependent diabetics, there's a reduction in GSK3 phosphorylation at submaximal insulin concentrations. So in summary, there's evidence in human insulin-resistant muscle to be defects in the insulin signaling cascade all the way down to AKT. So the big question that many have been interested in, and what we're quite interested in, is, are these signaling defects a cause or a consequence of insulin resistance? And there's been a lot of work along those lines looking at IRS and PI3 kinase manipulations. We're going to look at sort of the most distal part of the pathway in today's series, really focused on AKT. And so what we know, we're quite interested in AKT for several reasons, but both mouse and human mutations in AKT suggest a critical role in glucose homeostasis. So the AKT2 knockout mouse, originally done by Maury Birnbaum, shown here, has a marked decrease in total glucose disposal, very similar to what I showed you before. And if you look at ex vivo glucose uptake, you can see at submaximal insulin concentrations, there's a reduction in insulin-stimulated 2-deoxyglucose uptake in AKT2 whole body knockout mice. In addition to these loss of function mouse studies, there's been some evidence in humans that AKT2 mutations lead to profound effects on insulin sensitivity and diabetes. So here is a loss of function study by looking at by George et al. Looking at AKT2 loss of function doing hyperinsulinemic clamps, and you can see on the x-axis here the proband insulin concentration of roughly 650 picomoles per liter has a really marked decrease in the insulin infusion rate. Again, this is a clamp study, less of an infusion, the more resistant an individual is. Higher infusion means the more sensitive an individual is to insulin. So loss of function in humans leads to insulin resistance, AKT2, and conversely, an activating mutation in AKT2 actually leads to hypoinsulinemic hypoglycemia. So these individuals, this paper by Hussein et al., the Science paper 2011, can show that individuals with activating AKT2 mutations have incredible enhancement of insulin sensitivity. So if you clamp these individuals, there is really a marked increase in the glucose infusion rate. Okay, so normally under these conditions, there really needs no infusion to maintain glycemia. However, these individuals need constant glucose infusion to avoid hypoglycemia. You can see glucose levels are below 40 mg per deciliter, quite low and risk for hypoglycemia, despite really undetectable insulin measurements consistent with a profound increase in insulin sensitivity. So for a long time, the studies have really focused on how AKT can drive this response. Predominantly, most of the beautiful work by David James and Maury Birnbaum and various others have focused on the adipocyte. The adipocyte, it became quite clear that AKT is required and sufficient to drive glucose uptake. And that primarily was afforded at the time because of the 3T3L1 system as such a marked insulin responsive cell type. There's been some work in the skeletal muscle to really indicate which is the primary roles. However, there's really no in vivo data at the moment to say that skeletal muscle specific AKT is absolutely required for this effect. So here's some data that's a recent paper, relatively recent in 2012 in Biochem Journal, that showed that AKT inhibitors, you can see nice dose dependently block insulin-stimulated glucose uptake in isolated rat soleus muscle. This effect with pharmacology, we can recapitulate in our own lab. It's quite clear if you acutely block AKT2 or AKT with small molecule inhibitors, you can affect ex vivo glucose uptake. Okay, so at this moment, the big question is that we have is can we really understand the role of AKT in an organism to really ask the question, what's the role for AKT in these processes? So again, this led to sort of this popular model for the molecular basis of insulin resistance. What I just showed you is that there's been clear indications of defects in AKT, in IRS, PI3 kinase, and AKT in insulin-resistant individuals. There is data from whole body knockouts in human studies that loss of AKT leads to insulin resistance. And there's been a lot of studies in fat cells and then some studies with pharmacology in isolated muscle that AKT is an important determinant of glucose transport and glucose uptake in skeletal muscle. So this led to this popular model for the molecular basis of insulin resistance. And this really suggests that a reduction in muscle AKT signaling is sufficient to lead to a reduction in GLUT4 translocation, thus causing hyperglycemia and the sequelae of insulin resistance. And this is supported by a lot of studies, really some pioneered by Jerry Schulman that has argued that as there's increase in obesity, there's increase in diacylglycerols, and that antagonizes insulin signaling at the level of IRS or more recently at the level of the insulin receptor. Alternatively, work by Scott Summers and others have argued that the increase in fat and fatty acids leads to the accumulation of ceramides, and ceramides can either directly inhibit AKT signaling or can activate phosphatases that are then dampen AKT signaling. Both of those hypotheses sort of impinge on AKT signaling in the skeletal muscle that is really supportive of this model of obesity-driven insulin resistance in humans. And if you look again, this is just a rodent study. If you look in the OB mouse, for instance, which it has extreme obesity and insulin resistance, you can see quite clearly that there's a reduction in glucose uptake at various concentrations of insulin. And then this is just a Western blot performed in my lab. If you could see at a level of AKT phosphorylation here indicated with serine 473, you can see this reduction in glucose uptake correlates with a decrease in AKT phosphorylation, sort of consistent with this model of reduced AKT leads to a reduction in glucose uptake. However, at the time, there has been no studies to my knowledge that has directly asked the question, is a reduction in skeletal muscle AKT alone both necessary and sufficient to cause insulin resistance? So that's exactly what my lab has embarked to do. And so to answer that question specifically, my lab has generated several skeletal muscle specific knockout animals of AKT deficiency. So for the aficionados, there's 3 AKT isoforms. There's AKT 1, 2, and 3. AKT 3 is predominantly expressed in the brain and the testes of both mice and humans, while AKT 1 is more ubiquitously expressed, and AKT 2 is highly enriched in metabolic tissues such as the muscle, liver, and adipocyte. So we began by making various individual knockouts first of AKT2 because, again, like I mentioned, its expression is most enriched in skeletal muscle, and then also in AKT1 because we have worked from our lab and previously when I was a postdoc and other work by Maury Birnbaum and others has argued that AKT1, at least in the liver, a little bit of AKT1 is sufficient to transmit sort of the insulin response to a lot of residual AKT activity. So we made both the AKT2 skeletal muscle specific knockout and then the AKT1/2 double knockout. We used here the human skeletal actin Cree, and you can see it's very specific for skeletal muscle, and of note, it does not affect cardiac tissue. So here's just a Western blot looking at AKT expression, either control or AKT muscle specific, skeletal muscle specific double knockout. Here's the AKT3 expression, really undetectable at the Western blot level of muscle, highly expressed in the brain, and is not upregulated in the absence of both AKT isoforms. And lastly, just to convince you that we're not affecting upstream signaling here, this is downstream AKT signaling. You can just inject insulin into a mouse, a control mouse, or a muscle-specific AKT knockout, and you can see a dramatic reduction in AKT phosphorylation, as you would expect, deleting the enzymes without affecting the phosphorylation status of the insulin receptor. Okay, so we have multiple mouse models here that we can really assess the isoform specificity of AKT. So the first thing you can obviously do when you're doing a skeletal muscle manipulation is you can first look at the muscle mass. And as you can see from this graph here on the left, AKT2 knockout alone in muscle really leads to no effects that we can see in terms of muscle mass. However, when we knock out both AKTs, you see a dramatic reduction in skeletal muscle weight. It's consistent with AKT being an anabolic signal. If we do the same experiment where we treat mice with insulin, either control AKT double knockout or AKT2 knockout, to really confirm if there's a defect in signaling, we did the classic sort of Western blot approach. And if you inject insulin in the AKT2 knockout, you can see a dramatic reduction in phosphorylated AKT. However, even despite a roughly 90% reduction in AKT phosphorylation, you can see downstream signaling to the canonical AKT targets, such as PRAS40 and mTOR, here marked serine 240 and 244, and 4-ABP, is preserved. However, in the AKT double knockout, you can see a complete absence of AKT phosphorylation and thus a corresponding absence of downstream signaling. So what this is telling us is that the residual amount of AKT1, roughly 10% of the AKT expression in muscle, is sufficient to transmit the insulin response. So there's a tremendous amount of spare capacity in AKT signaling that is available in skeletal muscle. And what I'm not showing you here is the AKT1 knockout alone, which was originally published using a different Cree, which included the cardiac excision. We did it with the HSA, and we also see no difference in signaling. So this is not an AKT1 specific effect. This is due to the residual amount of AKT expression in the muscle. So to be clear, a 90%, at least genetically, a reduction of 90% of the AKT is not sufficient to lead to a defect in insulin signaling downstream of AKT in muscle. Consistent with the AKT2 knockout having no effect on downstream signaling, AKT2 knockout mice alone have really normal glucose tolerance test and insulin tolerance test. So really no observable metabolic, or then you can see sort of the histological phenotype that we could observe. And that in comparison to the AKT double knockout, we see modest effects on glucose tolerance, however, fairly normal insulin tolerance. So we were kind of struck by this. You know, the insulin tolerance seemed quite normal. And again, the hypothesis is that AKT is absolutely required for glucose transport in muscle, yet the ITT looked quite normal. So we took the next step and did hyperinsulinemic euglycemic clamp studies in these AKT knockout mice and control mice. And as you can see in the left and the right, whole body glucose infusion rate were normal relative to control, and so was total glucose disposal. And this is data that's normalized to the lean muscle. So we did MRIs before the clamp studies, and so we're normalizing to the reduction in muscle mass because, as I mentioned to you, there is this defect in muscle mass. And at the end of the experiment, you could do the 2-deoxyglucose uptake labeling, and you can see both in the soleus CDL and the gastrocnemius muscle, in vivo 2-deoxy C14 glucose uptake was normal in mice that lacked AKT signaling, specifically in the skeletal muscle. We obviously were surprised by this finding, so we wanted to make sure we can confirm this in ex vivo muscle, where we've isolated muscle, both soleus and EDL. Here I'm showing you the soleus muscle, and performed ex vivo insulin-stimulated glucose uptake. And you can see in the control conditions, you have a nice increase with insulin that is suppressible by PI3 kinase inhibition. In the AKT knockout, a similar effect is seen, so you can see an increase in insulin-stimulated glucose uptake. Again, that is suppressible by PI3 kinase inhibition, as indicated by the Ward-Manin experiment. So in conclusion, AKT deficient muscle has normal rates of insulin-stimulated glucose uptake, both in vivo and then ex vivo. And so obviously, we wanted to really investigate mechanistically how this was happening. And a lot of the work, primarily again in the adipocytes, most of this really elegant work has been done, has implicated, but there is some in the muscle that has implicated the role of RAB gaps, or in particular TPC1D1 and 4, in controlling RAB GTPase activity and GLUT4 translocation in various situations. So we first looked at the AS160 signaling. This is a canonical, or at least implicated at the time to be a canonical AKT target. So here you can see increase in phosphorylation status of AS160 at 642. And this indeed is an AKT site. In the AKT knockout, we see no increase in insulin-stimulated AS160 phosphorylation at this residue, consistent with this being an AKT substrate. If you do a pan AKT substrate Western blot for that site around, this is a phospho-AKT substrate blot 160, just cropped at that 160 kilodalton, you can see, again, no increase. We don't have good antibodies ourselves to do the TBC1D phosphorylation. We're working on that, but if you look at total TBC1D1 levels, they look to be somewhat suppressed, which is consistent with recent studies by Brian O'Neill and from Ron Kahn's lab, and now in his own laboratory, that has indicated that TBC1D1 protein expression is decreased in insulin receptor IGF-1 receptor double knockout skeletal muscle. These defects, so this lack of AS160 phosphorylation would argue that this site is not required, at least acute activation of this site is not required for insulin-stimulated glucose uptake, as I just mentioned to you, as I just showed you. And if you do crude plasma membrane fractionation studies, you can see relatively similar levels of GLUT4 and GLUT1 at the plasma membrane in insulin-injected AKT knockout mice. By no means is that a translocation assay, but just says in general, after an insulin response, there's similar levels to GLUT1, GLUT4. So this is sort of the traditional view. that insulin stimulates glucose uptake via an AKT pathway. And this goes through TBC1D1 to stimulate GLUT4, leading to various distal processes involved in actin remodeling, RAC activation, et cetera, which I'm not going to focus on here, that leads to GLUT4 translocation and thus glucose uptake. What these data I think would indicate is that, at least in skeletal muscle, now I'm focusing here on skeletal muscle, that there has to be an AKT-independent pathway that can lead to GLUT4 translocation that sort of seems to be independent of the canonical AKT TBC1D1/4 pathway to promote glucose uptake via GLUT4 translocation. And so obviously, anytime you do genetic experiments, especially from a congenital knockout of birth using this approach, we were very concerned initially that this was just some compensatory pathway that was activated due to developmental compensation from lack of AKT. And fortunately, there's Cree models that have been developed that has allowed us to address that from Messer colleagues. Here is a similar promoter, human skeletal actin, but now this is under the control of the estrogen receptor, so we can do a tamoxifen inducible system. So just like before, we generated several AKT-specific flox mice, crossed them with the Cree, the inducible Cree, to do the same experiment here. But the difference is we let these mice become adults, roughly 8 to 12 weeks of age, and then treat them with tamoxifen, and then look acutely. Now we can look temporally here. We can look at 2 weeks, 4 weeks, whenever we get excision, to assess the role of AKT in insulin sensitivity in the developed adult muscle. So we took that same approach. Again, I'm just going to focus on the AKT double knockout here. When we did this with the AKT2 knockout, again, the AKT2 makes up most of the AKT in skeletal muscle. However, we could not see any phenotype in terms of muscle mass or in terms of insulin sensitivity when just getting rid of AKT2. And more striking was the fact that we did not, despite deleting 85 to 90% of the total AKT pool by getting rid of AKT2, that did not affect downstream signaling, which is sort of consistent with work by David James from years ago that has argued that there's a tremendous amount of spare capacity in the AKT signaling both in muscle and in adipocyte. So using this approach, you can see we can get rid of AKT2 here. And then this has really, at least at 4 weeks here, this is looking post-tamoxifen. You can see minimal effects on muscle wasting. You can see the gastrocnemius muscle, it's a little bit more dramatic. And this is due to this sort of acute nature of this. So we can try to use this model where we don't have this profound muscle atrophy to really get at what's the role of AKT signaling in this context. Of note, if you let these mice, if you go 8 to 12 weeks following tamoxifen, you do see the more profound atrophy, which will become relevant in the second half of this presentation. But at least acutely, you really can minimize the secondary loss of muscle mass due to AKT deficiency. We do very similar experiments. We first assessed AKT signaling in these inducible AKT knockouts. Sorry, this, for whatever reason, labeling didn't come through here, but this is minus or plus insulin treatment, again, in vivo injection. You can see insulin injection leads to robust activation of AKT in control muscle, as indicated by the pan-AKT phosphoantibody or the AKT2 phosphospecific antibody. In the AKT double knockout inducible mouse, you can see this is completely lost, and there's lack of activation of Pres40, for instance, and mTOR as good surrogates of AKT signaling. If you perform a similar insulin tolerance test 4 weeks post-tamoxifen injection, you can see a very similar, recapitulating the congenital phenotype, acute deletion of AKT at 4 weeks leads to apparently no abnormalities in insulin sensitivity in mice. This effect, the autonomous effect on insulin signaling in terms of glucose uptake, was confirmed ex vivo. Very similar experiment was performed in the congenital knockout. You can see insulin stimulated leads to robust glucose uptake. Here, that's suppressible by PI3 kinase inhibition. In the inducible AKT knockout, you can see a similar effect on insulin stimulated glucose uptake. Again, that is suppressible by PI3 kinase inhibition. And same story as the congenital. The insulin leads to AS160 phosphorylation. We do not see AS160 phosphorylation in double knockout in the inducible form. And this leads, again, we can see similar levels of GLUT4 and GLUT1 at the membrane in these mice. So collectively, we really don't think that this is just a developmental issue. This seems to be when you have chronic AKT deficiency, either genetically, there must be a pathway that exists to downstream of PI3 kinase, right? Because all the experiments I showed to you with PI3 kinase inhibitor Wart-Mannin would block this effect. So this is a PI3 kinase dependent effect that would lead to GLUT4 translocation in this parallel AKT pathway. Importantly, the big question is this relevant to obesity-induced insulin resistance, or is this just some genetic condition where if you get rid of AKT completely, you've unraveled this new pathway? I mean, as a scientist, I think that's quite interesting, the fact that a pathway can exist, but for us to really get that excited about it, this has to be relevant in a physiological context, not just AKT deficiency. Fortunately, most of us are not walking around with complete loss of AKT in our skeletal muscles, where we would have quite a difficult time. So we can ask a simple question: is this insulin-dependent AKT-independent pathway sensitive to obesity-induced insulin resistance? Right? If the model, again, the conceptual model is if it's all about AKT deficiency, you should be able to put these mice on a high-fat diet, for instance. That should, in theory, block AKT and thus have a defect glucose uptake. If you did that in an AKT deficient animal, you would predict that the obesity situation would have no additive effect because we're already recapitulating that in a genetic effect. On the other hand, if those obesogenic conditions has an effect on insulin sensitivity on the double knockout, that would give a lot of support to the physiological relevance that there has to be a pathway in addition to AKT in muscle that's important for glucose uptake. So we did that experiment and we put mice on 12 weeks of high-fat diet feeding and did very similar experiments. And as you could see, looking at 12 weeks, unlike on normal chow, the AKT double knockout mice are more insulin resistant compared to a control. Again, this was a pilot experiment at the time. Unfortunately, the control conditions were happening. This was an initial pilot on the high-fat diet. The control conditions are happening right now. Unfortunately, as many of you are enduring during this shutdown, we were unable to complete that control part of the experiment, but hopefully when we get back up and running, the controls will be here. But if you remember what I showed you initially, same age groups, under control feeding, the high-fat diet, the AKT knockouts had normal insulin sensitivity. Here, when you give them high-fat diet, they seem to be more sensitive to high-fat diet in terms of insulin resistance. And then if you isolate muscle and stimulate with insulin, if you stimulate with insulin to measure glucose uptake, high-fat diet has a modest effect. Again, if I showed you controls here, remember it's up 2-3-fold. So there is an effect in control. However, the double knockout under these conditions is really unable to stimulate glucose uptake. So that led us to really hypothesize that there's some pathway that's here antagonized by under these conditions to block glucose uptake. So what is the pathway? We don't have it all worked out, but I think we have some clues about what's going on. So big question again is, what is this insulin-dependent AKT-independent pathway? And we turned actually initially to AMP kinase. AMP kinase has been historically implicated to be a pretty important regulator of glucose transport in skeletal muscle. It's been, especially recently, it's been the target of some pharmacology to see if you can stimulate AMPK and if that's an effective way to promote glucose uptake. Classic studies, which I'm going to show you a couple, have implicated AMPK activation in response to exercise and has argued that has led to improved insulin sensitivity. So given some of these studies and just kind of walk through these, so AMPK is this kinase that's been involved and implicated for GLUT4 translocation for years. It's known to be activated in response to exercise. So this is a classic experiment done by Hardy et al. In the 90s. They showed if you exercise mice, you can see an increase in AMPK activity as we get on the top here, and corresponding decrease in its target ACC activity as you run mice on the x-axis. Work by Pfizer, here I'm highlighting from Russell Miller's group, has argued that if you pharmacologically activate AMPK, people have done this with ACAR, they have a more selective compound. You can see a nice increase in glucose uptake in skeletal muscle. That is then blocked if you obviously delete both of these, if you delete the AMPK isoforms. And so there's certain situations. So clearly activating AMPK can promote glucose uptake. But the question is here is there any condition where AMPK is responsive to insulin, right? Because these are insulin stimulated conditions. And it's known for quite a while that exercise can affect muscle insulin sensitivity. So here is work from Richter in the early 80s and JCI, showing that if you exercise rodents and then perform insulin stimulated glucose uptake in skeletal muscle, you can see a nice dose response shift to the left. So exercise muscle becomes more sensitive to insulin. So if you exercise and do an insulin stimulated glucose uptake a test, you can see a dose response shift to the left. And work here, there's been several studies, but here I'm just highlighting from Bobstead et al. Recent diabetes people have argued that increase in insulin sensitivity following exercise is dependent upon AMPK. And then it's also independent of AKT. So highlight here on the bottom, here's this AKT phosphorylation, either in contracted muscle or rested muscle. So rest is in the open, contracted is in the black. And you can see there's no effects on insulin sensitivity on AKT phosphorylation irrespective of exercise. However, as shown here, you can see that the increase if you prior contract a muscle and then stimulate with insulin, you see a nice 50% increase in glucose uptake. This response is dependent on AMPK activity. You do the same experiment in mice that lack skeletal muscle specific AMPK, you can see this effect is lost. So under certain conditions, AMPK can be a regulator of muscle insulin sensitivity. And some other work, that is really in cell models has argued that in response to prolonged serum starvation, insulin can activate AMPK, as shown here, and that if you measure glucose uptake in L6 cells, you might get a modest effect, and that can be antagonized if you block AMPK. So we hypothesized that AKT, in AKT deficient muscle, AMPK was being activated, and maybe that was contributing to the insulin sensitizing effects. So first we looked at AMPK activity just as a surrogate here as threonine 172 on AMPK. And you can see an increase in the phosphorylation of AMPK in AKT deficient muscle, which corresponded to a decrease in ATP levels. Working with Joe Bauer and Tim Luongo, we performed mitochondrial respiration assays in isolated mitochondria, and we can show that consistent with increased AMPK activity, we can have a decrease in the O2 respiration rate in mitochondria isolated from AKT knockouts. In addition, we had a total loss of mitochondrial content as measured by TOM20, which correlated with a decrease in PGC1 alpha and beta expression. So it appears chronic AKT deficiency in skeletal muscle induces mitochondrial dysfunction, and this correlates with an increased AMPK activity. So we hypothesized that AMPK was playing a role in these insulin sensitizing effects as acting as a parallel pathway to AKT. And so to test that hypothesis, we just have done some, done some initial experiments with pharmacology. We're making the genetic knockout animals soon. But here we can treat with compound C, a well-known, relatively specific AMPK inhibitor, and then a recently described ULK inhibitor that has been shown now more recently to be a more actually potent and specific inhibitor to compound C for AMPK, this SBI compound. And as work that's been demonstrated for years, inhibition of AMPK here with two distinct small molecules has no effect on control muscle, in terms of insulin stimulated glucose uptake. However, muscles derived from the AKT double knockout, muscle-specific AKT double knockout, are exquisitely sensitive to both AMPK inhibition, either with the compound C or with the SBI compound. So again, two different chemically distinct AMPK inhibitors has the same effect on insulin sensitivity in the AKT double knockout. So In conclusion, for part one, and then we'll spend about 15 minutes on the unpublished data, as I promised, is that mice lacking AKT1 or AKT2 in skeletal muscle appear phenotypically normal. AKT2 knockout alone, despite reducing AKT phosphorylation, total AKT phosphorylation by about 90%, does not affect downstream signaling. So there's a lot of spare capacity that's involved in AKT. And I think when we study AKT, we need to make sure we're looking downstream of AKT when we're reporting changes in phosphorylation status to make sure that there's a functional decrease in AKT activity. Muscle AKT double knockout have normal insulin sensitivity, and suggesting that inhibition of AKT alone in muscle is not sufficient to cause metabolic disease or insulin resistance of the organism. This can happen independent of the canonical AKT AS160 642 mechanism. And then we have some evidence to suggest that there's mitochondrial dysfunction that leads to the activation of AMPK. And at least ex vivo, this is required for the insulin sensitizing effects in the absence of AKT. Okay, so most of this was published relatively recently, last October, in Molecular Metabolism. And so we sort of, in summary, come to this sort of this new model of potential the signaling mechanisms underlying insulin action in skeletal muscle. Here, the classic model has AKT both being required and sufficient for GLUT4 translocation and glucose uptake, while AMPK is activated by contraction in a separate parallel pathway. That's sort of this classic model, because if you inhibit AMPK, it does not affect this pathway. And at least acutely, if you inhibited AKT, it did not affect this pathway or PI3 kinase. But we think there's much more interplay going on here. What I've hoped to have demonstrated and suggested to you is that following chronic AKT deficiency, AMPK can become a dominant regulator of GLUT4 translocation in response to both exercise and insulin. And we're really interested in trying to understand, defining exactly how the proximal mechanisms of AMPK are regulated, and hope we can report some new data in the future for you guys. Okay, so as I promised, a little bit of unpublished data is using these models of AKT deficiency in skeletal muscle to really try to understand how AKT and more broadly how insulin and IGF, for instance, control muscle hypertrophy and function. And so it's been known for quite a while now from Walsh and David Glass's group that activation of AKT is a potent anabolic stimulus, and that has been shown here. I'm just highlighting one of their studies, where if you use a transgenic AKT, myristoylated AKT, and activated AKT in skeletal muscle, you can see a dramatic increase in the lean mass of a mouse, and also a increase in the cross-sectional area of a muscle here, staying with the laminin staining and quantified here in these graphs. Transgenic AKT skeletal muscle expressor has a dramatic increase in mean fiber size. So it's quite clear that AKT is a potent anabolic stimulus from these earlier studies. What I've shown you before is that the loss of AKT in muscle has the opposite effects, so really consistent with AKT being an anabolic regulator. And we felt like we could use this model to try to start getting at the question, what are the essential downstream mechanisms for AKT that control both hypertrophy and muscle function? And like I mentioned, work from David Glass and others have argued that TGF-beta-like ligands activate the SMAD pathway, and cytokines such as TNF all can impinge again on AKT signaling to lead to muscle atrophy and inhibit the hypertrophy response and induce mitochondrial function. And we suggest that we have this model where we can begin to dissect these pathways, because we can recapitulate some of these effects in our genetic system. So just to remind you, the AKT double knockout mice has a reduction in muscle in total body weight, but that corresponds to a reduction in muscle mass of all the depots, roughly a 30 to 40% reduction in muscle weight in various depots. I'm showing males here. This is also true in females. Consistent with the reduction in mass, there's a reduction in the CSA here. So the cross-sectional area is quantified here. There's about a 40 to 50% reduction in both the soleus and the EDL cross-sectional area. This is very consistent with, here's just the loss of function experiment. This is very consistent with the work by Glass and Walsh et al. That's to do with that overexpression and activation of AKT can drive hypertrophy. We then were interested in how AKT controls the muscle fiber type. And as you can see here with MHC staining, if you look at the soleus muscle on the left, you can see a loss of the MHC1 positive fibers and with a gaining of the MHC2A fibers. So consistent with looking at more of an oxidative muscle, consistent with the shift of an oxidative towards a more glycolytic fiber type. And again, if you look in the EDL, you can see loss of 2A, stained here in the green, to more of 2B, consisting of even a glycolytic muscle is kind of shifting towards a more of a glycolytic phenotype, if you will, to gaining more 2B fibers while losing 2A fibers. Consistent with the reduction in mass and the fiber type, there's a functional defect. So if you just measure grip strength, no surprise, AKT knockout have a defect in grip strength. If you put them on involuntary wheels, they really don't like to run. And if you force them to run on treadmills, they have a defect in both the maximal distance run and their time to exhaustion. Obviously, these mice have less muscle, and that corresponds to defects in exercise performance. To get at more of the muscle-specific physiology, we teamed up with Teji Khurana's laboratory at University of Pennsylvania and Emanuele Luro, while he was in Teji's lab, to perform ex vivo physiology assessment in skeletal muscle. And you can see both twitch and tetanic force of absolute force is decreased. Again, this is not a surprise. There's less muscle there, but if you normalize that data to the specific force, in general, most of the force that can be generated is normalized. So it seems that AKT's control of muscle strength is largely dependent on mass. There may be an effect here in the soleus, and we're still trying to tease apart. Okay, so what's downstream? Beautiful work primarily in cell models by David Glass. David Glass's laboratory has argued that both TOR and FOXO pathways can contribute to muscle growth, and has shown both in certain situations can drive muscle growth if you inhibit and antagonize AKT in the C2C12 model system. Recent work by Brian O'Neill, while he's at Ron Kahn's lab and now his own laboratory, has argued that FOXOs can drive muscle atrophy downstream of the insulin IGF-1 receptor. So we wanted to really take a genetic approach and ask the question, if we activate TOR or inhibit FOXO pathways, were they sufficient to drive muscle growth in the absence of AKT? If so, we can define which mechanisms are the key to contributing to this phenotype. So we generated another series of genetic knockout animals. Here we generated an AKT FOXO1 triple knockout, or an AKT TSC triple knockout. So the TSC here, remember, TSC is a negative regulator of mTOR. So if you knock out TSC, you can activate TOR independent of upstream AKT signaling. This is a strategy we used in the liver before, and we're doing it here in the skeletal muscle. And again, FOXO is one of the predominant isoforms, FOXO1 and FOXO3 in skeletal muscle. If you look at the effects on muscle weight, you can see that there's a modest increase in muscle mass. However, it was not sufficient. So you get a little bit of an increase both with either FOXO1, additive deletion, or a TSC deletion. But if you look at the gastrocnemius, it's a little bit of a bigger muscle, you can see it's not quite sufficient to get you back up to normal levels. And what we then hypothesized is that, well, maybe it has to be a combination of both. If you just drive the TOR effect without inhibiting FOXO, you may be maybe not fully able to recapitulate what AKT is doing, right? So we need to do both at the same time. And so we generated this next mouse model, which is a sort of a quadruple knockout mouse, where we're deleting TSC and FOXO1 in addition to both AKT isoforms, and we were struck by the findings. When we generated this combination knockout, we can see we can have unrestrained increase in muscle mass. We have more muscle mass than control, despite no AKT. And this was true for all fiber types we looked at. Normalization of the body weight. And as you can tell from this, it looked to us initially, and I'll show you in a second, it looked to be more red fiber enriched in each depot, probably consistent with more of an oxidative type fiber, which we're going to verify in a second. In addition, the CSA for both the soleus and EDL was dramatically increased. You can see the increase in the CSA compared to both, frankly, the control, but obviously compared to the double knockout, consistent with not only is the muscle weight increasing, but so is the CSA. And as I mentioned, the red fiber type kind of, initially thought to us that there was a fiber type switch, and you can see staining here in the green. You can see obviously the fibers are larger, but there's an enhancement of the MHC type 1 fibers here being stained. This correlated to a normalization in the mitochondrial complexes, and TOM20, which I showed you before in novel knockout, was deficient. Normalization of the PGC1 expressor, and not the best blot, but the preliminary blot showed that the quadruple knockout no longer had AMPK activation, unlike the double knockout. Consistent with that, we're restoring the mitochondrial content and function despite lack of AKT. So both of these pathways were driving this phenotype. And then, most strikingly, this combined activation of TOR and FOXO led to increased muscle performance. This is just a crude metric here, but we did some extra treadmill running, and you can see, unlike double knockouts, most of the maximal distance run and time to exhaustion is largely improved in this quadruple knockout mouse. So in summary, for part 2, this unpublished portion is, I hope to have demonstrated to you all that mice lacking both AKTs have a reduction in muscle mass. This correlates with a reduction in the oxidative properties of skeletal muscle and subsequent muscle strength and exercise performance, and that the activation of TOR and inhibition of FOXO1 in particular are both required and sufficient to drive muscle mass, mitochondrial function, and exercise performance downstream of AKT. So collectively, as a cartoon illustrating this, I think this is very supportive of some initial work by David Glass using really cell approaches that has argued that both TOR and FOXO pathway are required and sufficient to control the effects of skeletal muscle growth and function downstream of AKT. So with that, I just want to thank everybody in the lab here. This is when we were actually allowed into the lab. Most of this work was led by a talented postdoc in my group, Natasha Jaswal, and with support from a research technician, Matthew Gavin. The clamp studies were done in collaboration with Joe Bauer and Will Quinn at the Phenotyping Core at the University of Pennsylvania, and the physiology assessments in the second part were done in collaboration with Emanuele Loro and Teji Karana from the Physiology Department of the University of Pennsylvania. We would not be able to do any of these studies without support from the National Institutes of Health, in particular the NIDDK, and with institutional money and support from the Perelman School of Medicine and the Institute for Diabetes, Obesity, and Metabolism at Penn. So with that, again, I thank everyone for spending the hour with me and learning a little bit about muscle physiology. I'm looking forward to all your questions, comments, critiques, concerns. On Twitter, I'll be available. Fortunately, my daughter fell asleep right next to me in the middle of it. There was only a little bit of pinching underneath the table during this talk that I'll be happy to answer. And with that, everyone, please stay well. And I look forward to meeting you all and seeing you all in person in the near future. Okay, take care all. Thanks.